Hello, I'm Hayley McDonald and my dissertation is on the effect of biphenyl A and polyvinyl chloride leachates on phytoplankton cell density. Here I'm referred to as BPA and PVC. 10% of all waste produced is plastic, but plastic makes up 80% of the waste entering the marine environment, with up to 12.7 million metric tons entering the oceans each year globally. That's equivalent to the total weight of the entire human population. The effect of plastic on marine wildlife has been well documented over the last few decades, with the focus on larger, more visible organisms. For example, in 2005, an albatross was found with plastic in its stomach originating from a World War II plane struck down in 1944 over 9,600 kilometers away. But what makes plastic such a problem? Plastic has some key properties that make it so widely used. It's lightweight, durable, cheap to produce, and versatile. It can be as thin and flimsy as cling film to as thick and strong as doors and construction materials. But once discarded and in the marine environment, these advantages now make it a serious hazardous material. Lightweight means it's buoyant, allowing it to be carried by currents around the world and be mistaken by wildlife as food or shelter. Durable becomes non-biodegradable. All plastic that has been produced, unless incinerated, remains unmineralized in pieces as small as 20 micrometers. Cheap means it's mass reproducible. A major portion of plastic is used for disposable or short-lived products that are discarded within 1 year of manufacture. And versatile means it's in high demand and is constantly being produced at a high rate. There are many types of plastics. These include the ones you can see here. These contain additives which are used to enhance the properties needed for production. There are many additives used, ranging from OPAs to phthalates and BPA. These help stabilize polymers, increase tensile strength, and make plastic more flexible, amongst other things. But these additives leach out over time to their surrounding environment. They've been found to cause significant consequences to organisms, including toxicity. BPA and PVC were chosen for this study, but why? BPA is used in resins on things ranging from plastic bottles to medical devices. In animals, BPA effects include sexual reproduction, development, and other processes regulated by the same hormones. Concentrations of up to 20 micrograms per litre have been found in the world's oceans. PVC PVC is a plastic containing additives. It's used widely from flooring to toys to medical equipment, with up to 25% of plastic used is PVC. In animals, it has high toxicity, has permanent oxidative stress, affects respiration, and overall survival decline. Of all plastic produced, 19% is PVC. Now we know why BPA and PVC was selected, but why was phytoplankton? Phytoplankton are one of the key species of the planet. They have emitted most of the oxygen in the atmosphere today, and they regulate CO2 levels in the ocean, controlling ocean acidification. They also remove carbon dioxide from the atmosphere as well. They're primary producers in the marine food web. They directly or indirectly affect all marine organisms. For example, all fish in their larval stage consume zooplankton, which in turn consume phytoplankton. Phytoplankton are also good indicators of climate change in the marine environment. This is because they aren't commercially exploited, so any long-term changes can be attributed to climate change. They're short-lived, so population size is less influenced by the persistence of previous generations, so there's a tight interaction between environmental change and planet dynamics. And their free-floating nature means they respond to changes easily by expanding or contracting their ranges. The two species used in this study are Amphidineum klebsi and Pyrocystis lunula. They're both dinoflagellates, which are most commonly known as the source of red tides. Pyrocystis are bioluminescent, and both species are found in the Baltic. Here they're important water quality indicators, and they're also used to monitor the effects of climate change. This is one reason why it's important to understand the effects of plastic leachate on these species. What are my hypotheses? I believe that BPA will have a negative impact on the cell density of phytoplankton populations. I also believe that PVC will also have a negative effect. I believe that when BPA and PVC are both used, there will be a larger effect on the populations exposed. But the method. So, the two species are used to see if there is a species-dependent effect. BPA solutions of 1, 5, 10 and 20 milligrams per litre are used, and PVC solutions of 0.25, 0.5, 1 and 2% are used. And these are made by allowing 5 grams of PVC cling film to leach into 100 ml of salt water over 8 days. All solutions are in a salt mix representing ocean salinity with an F2 fertilizer. 1 ml is removed each day to count the number of alive and dead cells over a 3-day period, and trypan blue is added to distinguish between the live and dead cells. What are the preliminary results? Well, here there are the results for the BPA. As you can clearly see, the specific growth rate of both species are significantly affected. As the concentrations of BPA increase, the specific growth rates decrease. But this effect was stronger in the Pyrocystis species, with every concentration having a negative specific growth rate. Whereas the Amphidaneem species, the 1% concentration still has a positive growth rate. But this still shows that both species are sensitive to having BPA in their surrounding environment. To continue with this, the experiment will be run with PVC and also combinations of BPA and PVC. It will also be run with a chlorophyll photospectrometer to get accurate cell counts. But what do the preliminary results mean? It means that for BPA, the hypothesis has seemingly been confirmed, and this means that the continued and increasing presence of BPA in the marine environment could have detrimental effects on phytoplankton populations. There could also be knock-on effects to all marine species, and it will also impact the carbon cycle. It would also result in the loss of a valuable tool used in climate change. Thank you for listening. Any questions?